In this video, we're going to learn about enzyme naming, where enzymes get their names from, and also how we can take an enzyme name and glean a lot of valuable information from the name itself. Now, we're going to notice that our enzymes usually have the ending ASE. So let's even write that down. Enzymes usually end in ASE. So when we see ASE at the end of the name, we can be pretty certain it's probably an enzyme. The rest of the name will identify the substrate. So who it's acting on. So ASE means enzyme. The rest of the name will talk about the substrate and also the enzyme function. In other words, like what kind of reactions is it going to undergo? Is reacting with that enzyme gonna do? So when we're talking about function, we're often talking about the types of reactions. So you know what? I'm going to put RXN in parentheses. I'm talking about like what reactions is it going to promote? Okay, so let's look at our first example, glucose oxidase. Okay, our ACE is really, our ASE is our clue that we've got an enzyme here. So we know we've got an enzyme from ASE. Then we can start from the beginning of the name. Glucose tells us the substrate. This is going to act on glucose. Now, since it's really specific to this one glucose molecule, that is probably going to be lock and key. I mean, we can deduce this. It's going to be specific, so it's a lock and key. And then it says it's glucose oxidase. So take a second. What reaction do you think this is going to be? Yeah, that's it. It's oxidation, right? So this is going to undergo oxidation. So it's going to do oxidation on glucose with a lock and key, type of specificity. Okay, let's try cytochrome oxidase. Okay, so the ASE tells us we've got our enzyme. Who is our substrate? Our substrate is something called cytochrome. Now, cytochrome is something in plants for photosynthesis. I'll just make a note there. Plants, cytochrome in photosynthesis. Now, this is really specific to the molecule cytochrome, so that's going to be a lock and key model. And then what's the reaction type? Well, it says oxidase again. So again, I'm going to just abbreviate it, OX. But again, we're doing oxidation here. Okay, so these first two are both oxidation of a specific molecule. So lock and key. Let's do sucrase. ASE ending tells us that it's an enzyme. Our substrate, you guessed it, sucrose. Again, that's one specific molecule. So that's going to be another lock and key mechanism. It's not a whole class of molecules. And this is going to actually be hydrolysis. So hydrolysis of sucrose, you've got a disaccharide. So you can split that disaccharide by hydrolysis. So that's what you're going to do with sucrase. Okay, how about methemoglobin? It looks like METH, like it's the prefix meth for one carbon, but it's about hemoglobin, so it's actually called methemoglobin. So we've got methemoglobin reductase. So it ends in ASE. It's an enzyme. It's going to act on the substrate methemoglobin. Since it's really specific like that, this is another lock and key. And then it says reductase. So that means it's undergoing a reduction reaction. So methemoglobin reductase is for a reduction reaction involving the specific compound methemoglobin. Okay, what about lipase? We've got the ASE ending for an enzyme. Lipase is interesting. It's going to act on the whole class of lipids. All right, so lipids is really broad, right? This is a broad class of molecules, so this is going to require an induced fit. This is going to require the enzyme to change shape depending on the length of chain of the lipid, for example. And lipids can undergo hydrolysis. That's really what we're talking about when we're talking about breaking down lipid chains, is we're breaking them down via hydrolysis. Okay, how about protease? So again, ASE enzyme, this is going to act on proteins. So we're just kind of going by what the name sounds like. This is another induced fit because proteins are a large class of molecules. And again, these are long chains that can be broken by hydrolysis. So, sucrase, a disaccharide, lipase, protease, we're talking about things that are, put together, right, in a disaccharide, in a triglyceride, in a protein, which is a long polymer of amino acids. So how are we going to break these things down? With hydrolysis, by adding water across, whatever we're trying to, whichever bond we're trying to break. Typically those ester linkages, for example. So doing hydrolysis, adding water across those ester linkages. Okay, we've got lactate dehydrogenase. Okay, so we've got our ASE ending. And then this is going to act on the substrate lactate. That's really specific, so that one will be lock and key. And much like it sounds, this is for something called dehydrogenation. We learned about hydrogenation, where you could add hydrogen across, say, double bonds. So dehydrogenation is going to be the opposite. It's going to actually be loss of hydrogens. Loss of hydrogens is actually, does oxidation in the process. So why it's not called lactate oxidation, lactase oxidase, that remains to be seen. I think it's because it's the direct removal of hydrogens versus the addition of oxygens, but it's the direct removal of hydrogens, which is technically an oxidation, but in organic, we like to reserve that word for only adding oxygens. So anyway, a little bit of semantics or details we don't need to get super mixed up in. So that one was really specific, lock and key. Okay, how about pancreatin? So this is going to act on lipids. Pancreatin is actually a mix of digestive enzymes. So proteins, amylase, lipase, lactase. So suffice it to say, this is induced fit because it's made up of such a mix. Of lipids that it can act on. So pancreatin itself is this mix of digestive enzymes, that therefore act on a broad class of lipids, and they're going to break them down. So it's going to be hydrolysis. So suffice it to say, pancreatin is made up of some of the prior, enzymes that we've seen, but those enzymes, things like lipase, were already induced fit. So, this is like a broader class of induced fit enzymes. That's how I would think of it. Because things like protease and lipase were induced fit. So this is like a mix of induced fit that breaks these down by hydrolysis. Okay. How about bromelain? This is induced fit as well. And it acts on proteins. That one's harder to tell just from the name, so I probably wouldn't expect you to know that one just from the name. This is actually a specific protease. So this is sort of a subset or a specific protease, and it's undergoing hydrolysis, or a type of protease. So similarly, hydrolysis induced fit. And then what about pepsin? This is going to act on a peptide. So peptides are going to get broken down by hydrolysis. And since, peptides are a pretty broad class of molecules, we're going to call that induced fit as well. So we're looking at, does this enzyme act on a specific molecule? That would be lock and key. Does it act on a broad class of molecules like lipids or proteins? That would be induced fit. And in general, when we've got these dimers like sucrose, when we've got triglycerides, when we've got protein polymers, generally speaking, a lot of these enzymes are breaking those down with hydrolysis. Otherwise, we usually specify if it's an oxidase, so if it's undergoing oxidation, if it's a reductase undergoing reduction, or dehydrogenase, undergoing dehydrogenation or loss of hydrogens. So, usually we specify the reactions that aren't hydrolysis in the name of the enzyme itself. So look for ASE and you can be pretty sure you have an enzyme and then start analyzing the rest of the name to learn more about it.